Okay, I see like my Google Colab is still running, so I'm assuming like your all ones are running, still running too. Huh? Oh, but they didn't cancel. Okay, then that's fine. Okay, and is the step 4 is still running for you or still running? Okay, so we still have to wait here. We can while we are waiting, let's see. Okay, I'll give you option. I can, while we are waiting, either I can question answer or I can go over docking. Oh, yeah. Okay. Actually, I forgot one of the important thing. We started get to know about each other. We stopped at 10 people. So you should continue from whoever didn't tell about yourself. We should continue from there. And before that, there was a question outside which asked like, the question was like, we do computational biology for prediction, and then we go to the wet lab, and I want to answer it for everybody. It's actually the reason that we do computational biology is to reduce the workload on the experiment. It's actually doesn't go like we predict something, we go to the experiment, not that. My experimental collaborator came, and here is the secret to be a successful computational biologist. You have to work with your collaborator. You have to pick up a local problem. You have to work with somebody that's you can collaborate with. Okay. Unless you are developing new methods in computational biology, in the sense like new algorithm or something, then to be successful, you have to collaborate with somebody. So now let's say you are collaborating with somebody and they came and they said like, you know, I don't have the resources to do 300 experiments, but that's what I need to do next. What a computational biologist comes, they say like, okay, let me try to make a model. We'll make a model, and then that model probably will reduce the 300 experiments to a 5 experiment. I'm still not giving the answer because I still do not know how accurate my answer is. But what I can tell is like, if you do those 5 experiments, then you have a chance to get the right answer. Okay, so, and I will give you an example. This is actually, this paper is just coming out. It will come out. This is, so one of my collaborator at where I work, he came, he told me like, you know, we got a new protein and we do not know what is the function, but nowadays there are so many online server to predict the function. We put the protein in there, we couldn't find the function prediction, right? And we think it's a protease, that means it cuts down other protein. But to predict it's a protease, we have to do so many experiments. We have to put all different kind of proteins with this and see if it works. So what can we do? So I said like, well, okay. So they had a protein, which is actually a huge protein. It has a beta barrel among lipid. And then there's a little loop here, and then this goes through, and then it has a lots of domains here. And the new protein, somewhere here, they know it interacts here. That's the only information we had. I said, why don't we do some docking? Okay, so we did docking, and we said like, I know my docking give me 25 results. Out of the 25 results, I think like 3 or 4 of them showed that this protein is interacting with this part. Okay, so then I said, okay, well, let's look at what it's interacting. And whatever it was interacting, it had 3 residues. I forgot now, it's like histidine, arginine, and something else, 3 residues. And they are very close to each other in 3-dimensional space, right there. But if you In the sequence one, they are like 40 residues apart. Now, I don't know what about the other one. I don't remember. So it was very hard to guess, like this was residue number 11, this is residue number 51, that's residue number 100 or something. It's very hard to get the functional perspective from it. But when I said like, okay, so these three residues are very close. You go back to literature, well, there are actually 5 residues here. Okay, let's put X1, X, X2, X3. These 6 residues are very close, but if you look at the literature, these 3 residues are actually signature for serine protease. So, well, why don't we go do some mutation studies? So they mutated, these are the signature for serine, they mutated. So they did 3 experiments, and all 3 that showed, like if you mutate, the functionality decreases. So I reduced the burden from few hundred experiments to 3 experiments, right? But it was not possible for me to tell that it is a serine protease. All I know is like, if my binding results were true, and remember I got 25 binding poses, out of that 22 was not true, even though the binding was telling me like this is like the best one. The only reason I chose the 3 or 4, because my collaborator gave me an information, this protein cleaves this protein right here. So I took that information, I said like, out of all my bindings, what are the poses where this protein binds right exactly here? So that reduced me from 25 to roughly 4. And then out of the 4, we looked like what are the residues. One of them had a signature of serine protease. Then let's do an experiment. So that's the role of computational biology. It's not the role to predict. It's the role to reduce the phase space of the experimentalist. You're going to work together. And then by reducing, so now this paper is coming, right? It took few months. So I did the work for a month, they did the work for 2 months, now it's coming. So that you have to keep in mind. Okay. If I did not have all this information, if I would have just come and docked it, I would have 25 results, right? And then I would have taken the top 1, top 2 and design experiment on it. I wouldn't have gone anywhere because the top 1, top 2 were actually not the right answer. Okay. So that's why every step, the computational biologist, you take the information from literature, you take the information from your collaborator, and you filter out the results you are getting. Then the remaining results, you design an experiment, ask them to design an experiment on it. And then you do the experiment, you got the validation, then you move to the next step. So that should be the role of computational biology. It's not predict, because there's so much approximation goes on. We just don't know which one is correct, which is wrong. Yeah.
You have said that the protein was modified in the residues that interact with transmembrane protein.
No, it's not modified. What happens is this loop gets cleaved by this protein. From previous experiment, it was known it gets cleaved here. That's the knowledge we had.
What about the doctor of the gene that is responsible for that protein?
What about what?
What about doctor of the gene that is responsible for that protein?
So that was the experiment that was done before. So they not exactly knocking out. They did an RNA-seq, which I think Sabrina is going to go over. Or you went over to RNA-seq or? Yeah. So she will go over. So you can actually do that's differential expression. That means like you look at the differences of protein level expression in a diseased or in another model, and you look at the expression in a wild type, and you see which proteins or which genes are overexpressed, which is underexpressed. And that was the first information came, like this is one of the protein that the expression level was different. So that was the first clue. This must be, well, this possibly is the cleavage protein, like protease protein. So after that, we came to the like, okay, we got the indication it might be the protease, but how do we design an experiment to confirm it is the protease? And now, without the computational biology, you would have probably had to design 100 experiments, but we reduced it to 3.
Sounds more positive.
Yes. Yes. Yeah, so from now on, whatever the project you decide, you just keep that in mind. Okay, what you are doing, whatever step you are doing, you are reducing the possibility. And then from there, you either look for the literature or look for collaborator to come up with a definite solution. And then you move to the next step and so on. Okay, so I was giving, so if you are still waiting for, the MD results, either I can, well, we should go with the introduction. So let's say on this side, everybody gave introduction on this side? You didn't, okay. Go ahead, start.
Myself, Mohammad Shahadat Hossain, postgraduate student from Mohammad Shahadat Hossain, postgraduate student from Biochemistry and Molecular Biology, Shahjalal University of Science and Technology, Sylhet, Bangladesh. Early in my research career, I fascinated the jiggling and wiggling nature of the atom and macromolecules, and I want to explore more about structural biology. Thanks.
So on this side, yeah, let's start from you. Who is in down here?
Done.
Hello all, I'm Shatila Shahi. I have finished my honors from Biochemistry and Molecular Biology at Shahjalal University of Science and Technology. And currently I am a thesis student there, also completed my master's there. I started my exploring things actually from my 4th year project, which was on differential gene expression of possible, to find out possible biomarkers. Then I switched to I switched to slightly to immunoinformatics to predict the immunological functionalities because for a mutant, for example. And now I'm currently working on a metagenomics of HIV, which might be turned into immunoinformatics, but I'm not quite sure. Actually, we are exploring, so we are seeing that in which way it could go. And apart from that, I love coding, and that is one of my hobby. And I like to read books, a lot of books, not the academic one. And I'm also, I don't know, should I call myself that but I am also part-time developer. I launched a simple e-commerce website 2 months ago, which was online for about 30 days, as it was a free server. So it now is on hold. So I'm now trying to develop a website for in where I could share my research idea or something like that. So I don't know how far I will go, but I'm trying actually. So that's me. Thanks.
Assalamu alaikum. Myself, MD Shakil Arman. I have completed my graduation in biochemistry and molecular biology at Shahjalal University and Science and Technology. While I have completed my post-graduation there, I am also a thesis student. So in my undergraduate, I have completed project on plant genomics, and my research topic was plant stress physiology. So I explored different genes that might have played some role in the plant genetics of plant stress physiology, because we know that now climate change is a big global issue that are affecting our production of the crop, because the different kind of abiotic stresses which are generated due to the climate changing and global warming. So that was my project there. Currently, I am interested in sequence genome analysis and variant analysis. And while I am also thinking about starting a project about studying the adverse effect of mRNA vaccine, and while also doing project on variant of HIV. Thank you.
Assalamu alaikum. I am Roke Akhtar Muna. I have completed my honors from Department of Fisheries, University of Dhaka. Now I am doing MS in Fisheries Biology and Genetics branch in this department. I am doing my thesis in identification of bacterial diversity from healthy to different disease pattern in shrimp, specifically bacterial disease HPND and viral disease WSD. Frankly speaking, I was not interested in bioinformatics in metagenomics, but after I completed one of my session in 6 days RNA metagenomics and this one, I'm feeling interested in this part, and wish me luck that inshallah I will do better in this session. Thank you. Inshallah.
Hello, good morning, everyone. This is Mamun Al-Assad. I'm from Department of Microbiology, Jahangir University. First of all, I would like to thanks to CompBio Bangladesh 2023 for giving me an opportunity as well as selecting me. And of course, I also would like to thanks to all of the instructors, especially Shishir, Rahat, Sajjad, of course, Sir and Sabrina ma'am for having a patience on us. So we are very new about this. And of course, I'm a PhD fellow of Bangladesh Academy of Science, So as part of my PhD, I am working with AMR. AMR, that is antimicrobial resistant genes. So the title of my PhD is Interconnecting Transmission of Antimicrobial Resistant Genes in Human Animal in Bangladesh. So, AMR is one of the great problem in our public health. So it's a threat to us. So I'm here. So after identification genomic analysis is very important. This is right or wrong, my genes identification. So I'm here for that is already did yesterday, and let's have a look for the next what happening. Thank you.
Assalamu alaikum. Myself, Muhammad Mahmudul Islam. Basically, I'm from Noakhali Science and Technology University from Department of Pharmacy, where I have completed my B Pharm and M Pharm. And currently I'm working as an academician at this department. So before I come to this workshop training, my knowledge on Linux was very poor, I must say. But during the couple of days, I'm getting addicted to Linux. And I think this platform will encourage me, a student or an academician like me, to engage more or my contribution towards this field. And I would like to be part of this organization on this field so that I can enlighten my students also. And thank you for everything. Assalamu alaikum.
Hello everyone, I am Onik Mojumdar, undergraduate final year student from Genetic Engineering and Biotechnology Department, University of Dhaka. About my research projects, what I did was, one of my projects was developing an automated tool for non-invasive parental testing data analysis. And another project I have completed was comparative metabolomic analysis between Oryza coarctata and Oryza sativa. Currently, I am working on the roles of novel microRNAs in renal cell carcinoma as my final year project. About this CompBio, I am very happy to tell you that I am learning a lot of things here. And previously, I didn't know a lot of stuff. And in this environment, among these brilliant instructors and this diverse peer group, I am much benefited, and I am very grateful to CompBio for that. Thank you very much.
Hello, everyone. I am Dr. Nilima Barman. Currently, I am doing my PhD of Dhaka University at Genetic Engineering and Biotechnology Department, and also doing my job as a laboratory medicine specialist in Bardem General Hospital, which is one of the largest hospital of non-communicable disease in the Southeast Asia region. I do my MPhil in laboratory medicine, where I have done some research work, especially on biochemical parameter in non-communicable disease and also communicable disease, including multi-drug resistant tuberculosis, hypertension, diabetes, and so on. During my PhD, I done some SNP detection in non-communicable disease, and I have some bioinformatics project like MD docking and simulation genome sequencing project. As a medical professional, bioinformatics is a new area of gaining knowledge for me. We have not enough accustomed with bioinformatics, and I'm so much privileged to be here to know knowledge and accustom myself with bioinformatics. Thanks that I am to be here. Thanks all.
Hello everyone, I'm Tahmina Akhtar. I'm a master's student at Department of Genetic Engineering and Biotechnology at University of Dhaka. In my undergrad project, I work on the association of natural antisense transcript with particular diseases like cancer, neurodegenerative diseases, and diabetes. And in my master's thesis, I'm working on two projects actually. One is on the metabolic profiling of diabetic patients, and the other is on watching the co-expression of particular transcription factors and transporters on cervical cancer tissues. So that's about my research experience, and I'm really immensely thankful for arranging this program and giving me the opportunity to participate here and learning the unknown, unseen things and getting introduced into the world of Linux and the other bioinformatics tools. I was really worried, how do I handle the vast amount of data I'm getting from mass spectrometry and immunohistochemistry. Now I am little confident that I can manage, as I am learning a lot of things from here. So thanks to you guys all for arranging this program. That's it.
I am Khalid Shahriar. I am at my final year of graduation at University of Dhaka. My subject is genetic engineering and biotechnology. And about my project, it was about spatial transcriptomics. What I have done is looking for some association of long non-coding RNA in prostate cancer and measuring their expression. And in this program, actually that it is coming handy about the molecular dynamics, that it can come handy for my downstream analysis of that project. So actually I'm learning a lot. So the thing is, given my qualification, I am lucky, I am honored to participate in this workshop. Thank you.
Hello. Good afternoon, everyone. I am Aditya Shubhasachir Khan from North South University. I came to know about this amazing workshop from a supervisor of mine who recommended me to come here to learn about the workshop and all the things that are taught here. I would like to take this opportunity to thank our course instructors, Dr. Amitaba Roy, Sabrina Madam, for everything they taught us through this journey, and other organizers who really helped us a lot. Otherwise, these programs will not be successful. And there are many other hidden people whose names we don't know, but they have contributed enough, I guess. So I'm the outlier in this group. It's an honor and absolute privilege to be among these talented and exceptionally brilliant people. I am really honored to be here, and thank you everyone for tolerating me. I am a beginner. I'm just learning. Even 1 year ago, I didn't know anything about programming, bioinformatics, or computational biology. I am just learning and thinking of a better future for me and for ourselves. That's it. Thank you. That's me. Thank you.
Assalamu alaikum. I am Ivy Sultana from Department of Microbiology, Jahangirnagar University. I am currently working in a group collaboration, and their group name is Podda Bio Research. My research area is too small. I just worked on drug designing and vaccine designing. Now I want to explore myself, and this platform is a giving very big opportunity to me. So thank you for selecting me. Thank you.
Hello everyone, I'm Afsana Reshmi. I have graduated from the Department of Genetic Engineering and Biotechnology, University of Dhaka. Now I'm doing my master's in that department. In my 4th year project, I have worked on the epigenetic changes due to the heavy metal. And in my master's, I have not decided the thesis topic. In my undergraduate, I have done 2 online courses on the drug designing. I had a lot of questions there. In this session, I got so many answers from the session. Thank you all the instructor for arranging this wonderful session. Thank you, sir.
Hello, everyone. I am Kalpana Boiddo. I am a graduate student from the Department of Genetic Engineering and Biotechnology at University of Dhaka. So currently I am doing my master's and just started my thesis work. In my 4th year project, I have done designing a biofilter for car pipeline. As we all know, air pollution is the biggest threat in our environment, for our environment nowadays. So we have designed a biofilter. It's just a theoretical project. And currently I am doing my thesis work with a pathogenic bacterial strain, which was collected from UTI patients. We have already done the whole genome sequencing, and now I am here to learn how to assemble and annotate that data. So I would like to thank CompBio Bangladesh, which has created such a platform for beginners like us. And I would like to immensely thank all the instructors here for giving us this opportunity for our better future. Thank you.
Hello everyone. I am Jamilul Islam. I have completed my bachelor's from Department of Genetic Engineering and Biotechnology, University of Dhaka. Now I'm doing my master's here. I am working on population genetics as my master's thesis project. And I am working with different DNA from different species like cat, dog, and cow, etc. And I want to see the differences and similarity between those species. And this is all about my project. And thank you, everyone. And thank you, the organizer, for selecting me. That's all.
Hello, everyone. Assalamu alaikum and namaskar. My name is Sanjida Islam Dorin. I am from University of Dhaka, Department of Genetic Engineering and Biotechnology. I am working on SNPs screening, or single nucleotide polymorphisms related to breast cancer. And thank you for arranging such workshop. Thank you so much.
Okay. এখন তো good afternoon হয়নি, afternoon. I think almost everyone knows me now. I'm a faculty member at KUET. Special thanks to Dr. Amitabh sir for arranging such a good workshop. And I can still see the shining eyes, the brightening eyes of these students. And I'm confident that they're learning a lot. But to me, it was really quite hard to digest. So my field is to just identify the bacteria or even viruses in the wastewater. So I really didn't think of I can do something better than just identification. But after this workshop, now I think I can go beyond. and I'm looking for some collaboration with these students, particularly among, I mean, she's also working on heavy metal. So I have a publication also on heavy metal. I think now I can go to see the changes that this heavy metal can make at cellular level or DNA level. So again, thank you so much for arranging such a good workshop, and especially at KUET. Maybe next time we will see again. Yeah, thank you, sir.
Okay, thanks everyone. Actually, I like the idea that you should make some collaboration within yourselves. That will be really good. So I think I was supposed to go over MD analysis. I do not know where do we stand on the, let me see. Okay, so my Google collab is still running, but almost done, 96% done. So if anyone of you has completed, you know, I do not know, even somebody got completed the Google collab? Your one got completed, good. Step 5, yeah, so it's almost done. So I can talk a little bit about something else. And what about the MD trajectories? Are those still running? It's done? Okay, so if it is done, why don't you show how to download them? Let's download them.
So I think you can all see my screen, right? Okay, so if you're done with the MD simulation, so you have to go to the step 4 folder. So first check if you are in this directory. That is user PGS0299, your account name, CompBio, CharmGUI, and then NAMD. Yeah, it should be something like that. Okay, are you there yet? Okay, so now download the step 3 input .psf file by clicking here and then download. Yeah, this one. And also download this step 3 input.pdb. Yeah. Done? Okay. Now you have to go to the step 4 folder. Click on step 4. You'll see a file named step 4 _ equilibrium.dcd. This file. Also download this file. Yeah, in step 4. Check if you are seeing this directory. This file, this DCT file. Okay. So download it from here. Okay, so if you are done with the downloading, then you should see your all files in the downloads folder, I guess, if you're using Ubuntu or some other distro. You should able to see these 3 files in your computer, personal computer. Check if you have downloaded these 3 files. Let's see. How many of you have not find these 3 files in your laptop? Raise your hand. Oh, okay. Okay, so yeah.
Okay, so next part of the lecture will be on the blackboard or whiteboard. So now you are all going to have a trajectory file, right? In an MD simulation file. And you see, like, this is called equilibration. Okay, so this is actually not the file that you do your analysis on. You'll run what we call the next part is production. Okay, so, and I'll go through what it means. But you'll have a trajectory file that I think Shishir and Rahat are going to show how to analyze them on the board. So what is the difference between equilibration and the production? Okay, so when you start with a PDB file, you start with the structure either from AlphaFold or X-ray crystallography. If you start with an experiment, the first thing you know, almost all the experiments for structure building is done at -100 degrees, -30 degrees. They are not done in room temperature. It's very hard to contain a protein in the room temperature and get those detailed structures. And then the AlphaFold is taking the data from their PDB. So not necessarily the structure that you're going to start with, it is a relaxed structure in room temperature. Okay, so that's why the first step, you know, you get a structure. Okay, and the step 4 equilibration, what it does is it relaxes the structure, because now it was seeing ice here, right? Because either X-ray crystallography or something, you had some other materials, either ice or other chemicals here. And now we brought it in water, right? In my MD simulation, I have water. So this is my preparation, like step 3 up to step 3. Okay, step 4. So when you bring from step 3 to step 4, what you are taking an experimental structure, get it familiarized with the surroundings, like water and ions that you put. And that's why it's called equilibration. Okay. That's why this step's name is equilibration. And we'll see certain, analysis, how you know your system has been equilibrated. And once it is equilibrated, this is the step 4. The step 5 is the production. That means once the protein is happy with its surroundings, then you start your production run. That is the step 5. And that's where you do your final analysis on. And this protein will probably do something, all these things, whatever. So we are not running step 5 because you ran the step 4 for 1 hour. Step 5, depending on your program, you'll run for weeks or months or something. But it's very important to make sure everything is correct up to step 4 before you go to the production run, step 5. Okay. So once you convince yourself, you convince your peers, you convince the instructors that you are ready to run a long-range simulation, then you go to the step 5. Okay. Because those things are little computational resource. You need a lot of computational resources, and we have limited resources. So we should be careful. Okay. So now let's say you get a trajectory from step 5. What do you do next with it, right? So the first few things that you want to check, if your protein is stable, those are the first thing before analyzing anything. Like in the sense that if your protein is not stable, that means either there was some problem in the simulation setup or the protein is not in biologically relevant environment. Okay. So how do you check the protein is stable? The first one way to check it is called RMSD. Okay. So how many of you heard the term RMSD? Have you? Okay, few, good. What does it stand for? Okay, and then what, let's see, if we want to write RMSD is such a beautiful name because it gives all the hints in the name itself. Let's look at it. So first you calculate the deviation, right? So if you have a structure like this and another structure went like this, so deviation means like you pick up an atom, atom number 1, atom number 1, atom number 2, 2, let's say 3, 3, 4, 4, and 5. Let's say we have 5 atoms. You overlap into each other. So the deviation between these two should be 0. So deviation means, let's say, so this is time 0. Let's say we call it, or time 1. Time 1, this is time 2. So x1 at time step 1 of the first atom minus time step 1, time step 2 of the first atom. Okay, so that's the deviation. So we calculate the deviation. Then square, right? We square it. Okay, and then mean. That means we sum over all the atoms. So I change it to I. So I sum over I. Okay, and then I take the root. That means, mean means there will be a number divided by number. Yeah. So that's how you calculate deviation. Then you take the square, then you calculate 0. I put it 1, but typically it says the 0th structure. Or you have a separate structure you saved somewhere, you can use that too. Either way is fine. Okay, so that's your reference structure. Okay, so the time frame 1, you bring it, and let's say if I have 2 colors, I can superimpose the 2. So this is, let's say, my reference structure, either time step 0 or something I saved somewhere. So I'm going to bring, I have this structure here, and I'm going to bring this structure on top of that. So then it will be like something like that, right? I'm going to superimpose. Huh? So that's what it's going to do. So that's for time step, first time step, right? So I'm going to calculate for each atom. I'm going to calculate these values and then average. So I'll have a value, let's say 1.0. So for first time spot, I'll have a 1.0. Okay. And then next time step, I'm going to bring the second snapshot to my reference structure. I'll bring it over and I'll carry on the calculation. And I'm going to keep doing it. And typically, an RMSD, what you are going to see is like, it will start, that's RMSF, we'll come to that. Okay, so it will start at a value around 1 or 2, in the sense that, as I said, if a structure has an RMSD of 2 angstrom width, that means it's a very similar structure. You never get an RMSD of 0 as soon as you let it fluctuate. So you'll start around between 1 and 2. Let's put 1.5. And then initially, it will increasing. And this is, you are looking at the step 5, like the production. It will increase. And depending on how rigid or flexible your structure is, then it's going to plateau. Okay. And when it plateaus, that's an indication, first indication that your system has equilibrated. Now, but not yet, in the sense that if it plateaus around 5, 6, 7 angstrom, that's fine. But if it goes around 10, 12 angstrom, that means like your protein has moved away from your starting conformation. Now, it can happen for 2 reasons. It can happen for biological reason, or it can happen something was not right in your simulation. Okay, so if this one plateaus or not even plateaus, keep increasing. So that's the first sign your simulation, either for biological reason, that's the not right system, or you did something else. So that's the first indication you are going to see. But if it plateaus, then, typically, I don't know what you'll see, 5, 6, 7, okay, that's fine. Okay, so that's the first indication you're going to see, like, okay, is this, my simulation is okay. And sometimes if your protein is very rigid, that means like mostly beta sheet and not much loops and stuff, you can actually even see at 3 angstrom or something, it plateaus. Rigid proteins don't fluctuate much and so on. Okay, so that's one, that's the first thing that you look, RMSD. Well, not the first thing, one of the first things you look, okay. RMSD, okay. And that, remember, RMSD, you average over all the atoms at each time step. So your x-axis is time, y-axis is RMSD in angstrom. Typically in angstrom is the answers here. Okay, any question about RMSD? One thing you should remember, RMSD is not a linear function, in the sense that if 2 protein has a difference of 2 angstrom RMSD, and another protein has difference 4 angstrom, it doesn't mean that the 4 angstrom protein is twice as deviation from the 2 angstrom. That's why RMSD is a good tool, but not a super good tool, because it's not linear. It's hard to, it's like 8 angstrom is really bad, but 4 angstrom is not bad, right? It's not linear. Okay, so. Okay, so good question. So is there any, so let's say your RMSD kept increasing, right? So is there something is happening? So what are the times, like I have seen, when does the RMSD kept increasing? Let's say Okay, so let's say you started your simulation with 2 proteins. Okay, so let's put my 2 proteins here. Okay, and as I started simulation, the 2 proteins, they don't like each other. So they started being like there. Okay, so then you have to ask yourself, why did I start with these? So you must have some reason, right, that you started with these. If you are convinced that you have started with these and your proteins don't like each other, they're moving apart. So there's either you did something wrong. If you go through something wrong, you checked all the things, so nothing looks wrong. In that case means you did not, like what can be, and I'll come back, what can go wrong, okay? Like in your setup, how to check up. But let's say you looked in your setup and you said like, oh, nothing is wrong. That means that then you did not replicate the cellular system in your simulation. And what does it mean? Sometimes you need a cofactor. A cofactor means like you need a third molecule. Yeah, you need a third molecule. It can be an NADPH, it can be glycolization, it can be a lipid or sugar molecule. Sometimes you need a third molecule, fourth molecule to keep them bound together. And you didn't have that in your simulation. So they are not going to like each other. So, and that will tell you, and that, okay, and now after checking everything, even then you say, oh, I was right. And those 2 things just flies away. Then you have to start come back. It's like, who told me to put this here? There must be something like, then that's a real long path. Because if somebody already said like these 2 should be together, and if you want to prove somebody wrong, then you have to go through a little, more work. So when you say reference, what do you mean? Oh, no. Okay. Sorry, I didn't make it clear. So let's say you have the step 5. Whatever if you made your, setting frequency same for each snapshot. Oh, it is showing. For each snapshot, you'll have in the output file, value for a temperature, value for an energy, and value for a kinetic energy, and value for potential energy. Okay, and you should have other values for each, if you save it, right, for each. So the first thing you check is like time step versus temperature. Okay. Do you know what do you expect from this? Remember we asked it to 303 degree, 303 something, right? What does this plot should look like? Straight? Actually, it will look like, okay, with an average value of 300. The reason is temperature is not well defined for atomic system. Temperature is a macroscopic system. Only temperature is well defined when your system gets big enough. Okay. And but the average should be right there. Yeah. But if your temperature start rising, okay, then you have a problem, because we asked it to be a 303 degree. And nowadays, the charm GUI, those things are so good that you should not have that problem. But maybe you change something somewhere. So that's one real place to see, So that one place, first place. I used to look at that place, but nowadays nobody published that things are really good. That means your system hasn't found its native structure here. So you should not do any analysis on that. So these are the temperature versus potential energy, time versus potential energy, time versus RMSD. This will give you some indication like is your MD simulation system is right. Okay, so now you convinced yourself the simulation system is right. And now you come to the RMSD and suddenly you see a jump in RMSD, then you should be excited because now something biologically is happening. Okay, so after you convinced yourself and that everything is right, and now I see certain things, that means like some kind of loop is opening up, something is happening. So at that time, the RMSD value not only gives you the indication of the stability, it also gives you that something is happening. Okay, so those are the first places. And again, the analysis of MD, that's the hardest part. And there are so many different techniques and comes. But these are the first, if you're starting new, these are the first thing you should look. And then after that, once you can formulate your question, like what exactly I'm looking for, you'll design your analysis based on that. So we will go through, when I say we, our 3 student instructor, they'll go through all this. Okay, so the 2 other part of the analysis that we will go through, well, 3 actually. Okay, so the other part is called RMSF, root mean square fluctuation. And this is slightly different. What RMSF does, and this is not for stabilization, this is more for to check if you have a system, two systems, one is mutated and one is not mutated. Okay, if something is, that's the first place you look. What an RMSF does is, again, you have 1,000 snapshots, right? So in RMSD calculation, we are bringing one snapshot at a time on top of each other. At RMSF, you bring all the 1,000 on top of each other at the same time. Okay, so all of your snapshots are superimposed at the same time. And then you go by residue. Let's say I go, I have 1,000 different coordinates for these residues, right? And I calculate the root mean square deviation for those residues, but over 1,000, okay? So that's what I plot. So what should be on my x-axis now? So I put all the time together, okay? And I'm picking up one residue and I'm calculating the standard deviation of that residue. So in this one, I should have RMSF, right? I'm going to put the RMSF. What will be my x-axis? Residue number, right? So for each residue, residue 1, residue 2, and 3, and so on. Well, whatever number I have residue, right? Let's say 200 residues. So that's what I should have. So in this one, what you're going to see is like 1 angstrom. Let's see, 2 angstrom. And let's put something like 8 angstrom. So what you're going to see is like that, something like that, okay? So what it is like, anywhere you see something, your 1 angstrom, 2 angstrom, those are your alpha helical beta structures because they don't move much, right? And so you are going to see they are staying within 1, 2 angstrom. Wherever you have a flexible part, they move a lot. So you are going to see like 5, 6, 8 angstrom and so on. And this is the first place you come to see the effect of mutation. In the sense like you have a protein, and this will be a simple, like if you are lucky, the mutation has a very simple effect, and this is the first place. So you have a, let's say, protein wild type, and let's say I superimpose it with protein mutation 1, okay? And I see it will be very similar. Suddenly this one goes back up and this one. So that you can say like, this residue number 5 or whatever number it is. The mutation did that. So that's the first, simple effect that you are going to look for. Not necessarily you can see that not every mutation are so blunt. There are other mutations do work very subtly, but this is like first place that you're going to see. So remember RMSF is the fluctuation of residue averaged over time. So you have the x-axis is the residue number. RMSD is averaged over the residue. So the x-axis is you have the time snapshots. Okay. So these are the two. Okay. So now you have a mutation. Okay. So again, what kind of problem we can set up with an MD simulation, right? If somebody comes and tell me like, I have this mutation, I want to find what does it do, okay? Then I actually say like, sorry, I cannot answer. Do you have an experimental experiment set up where you saw something that happens in the mutation and you don't see something that didn't happen in the mutation? Only then I say like, okay, I can now try. Because of the time constraint in the MD simulation, it is very hard just to find like, oh, I have this mutation, what does it do? It's very hard. And a good question is, I have the mutation makes transportation of this metal, reduced transportation of this metal ion. The wild type has the metal ion transport, let's say good, the metal ion goes to the inside the nucleoside or something nuclear, but the mutation does not do. So now I have two things that I can compare. Okay, so that's the right question. That's That's more solid question to ask an MD simulation, right? So, so let's say, if my protein is here and a metal ion sits right here, and if I mutate this protein, and then this one gets that the metal ion cannot sit here, that you can simulate both the system, and you can compare the RMSF at this residue. You can compare the, where does the metal ion stay, and that will be a more definite answer. Okay, so in MD simulation, unless you are using some advanced techniques, the best place for the MD simulation where you compare two systems, because whatever the errors and uncertainty, they'll be the same in the both, and then you can like compare the two systems. Okay, so that's how you decide. So let's say something like this is not visible in the mutation. Okay, so where do we go to the next step? Okay, so the next step is hydrogen bond, because the hydrogen bond is the master of biology, right? I mean, like our room temperature at 300 degree Kelvin, the room temperature gives a, roughly 0.5 kilocalorie per joule energy. So a biological system, whenever it fluctuates, it can actually fluctuate within 0.5 kilocalorie per joule, kilocalorie per mole temperature energy. In that energy, that's the hydrogen bond energy fall in the range. So in the room temperature, the hydrogen bond can form and break. That's why all we are doing is hydrogen bond. In the room temperature, the ionic bonds, we cannot break them, right? I mean, so that's why in molecular dynamic simulation, you never see an ionic bond, because we are not putting extra energy and so on. Okay, so hydrogen bond is the next thing that you look for. And the way that you calculate hydrogen bond is actually, it's a little ad hoc, because there are so many hydrogen bonds and you cannot like track everything. The first way you do, the hydrogen bond, you have a donor, and then you have a donor, and then like, let's see, you see, my chemistry knowledge is not great. So electron, negative, so it's an acceptor, right? So hydrogen of another, so it's some kind of residue 1 and residue 2. So, okay, so this hydrogen atom has slightly positive charge. This oxygen atom in another residue has slightly negative charge. It can, so this one is called acceptor, because this is called donor, because it can donate an electron. And this will be called acceptor because it can accept an electron, right? So you will have a weak interaction between them. And that interaction is weak enough that it can form and break within a room temperature. But to identify that interaction, the way it's calculated, you only calculate, I think the bond length of hydrogen bond is probably around 2 angstrom, I forgot, maybe 2, 3, something like that. So you look at the distance between the two known donor, known acceptor, and the angle. So that's the analysis you first do. Like, does this donor atom, acceptor atom come within certain distance and has the right angle? If they have the right angle distance, then you say like, oh, they have a hydrogen bond, okay? And then if they do not have the right distance, then you do not have a possibility of creating a hydrogen bond. So that the next part of, again, the analysis they are going to show you on the board is like, you calculate all the possible hydrogen bonds and you see if the number of hydrogen bond has been reduced due to the mutation, or if the number of hydrogen bond has increased due to the mutation. So that means like if it has increased, certain part of the protein got more rigid or something. If it is decreased, the part. So that's like the next part you look into. Okay, so if your RMSF does not give you an answer, next you look at the hydrogen bond. In a lot of cases, I don't want to put a percentage, but in a huge number of cases, the hydrogen bond should be enough to, identify the effect of mutation. Like first you look at the total number of hydrogen bond, then you can go, you say like, I know like the hydrogen bond between this residue and this residue is the culprit due to the mutation. Then actually you can calculate the energy between these two or the distance between these two, and then you can follow. So hydrogen bond should be your next step to look into, okay? I think those three are most. There is another, and anything else require your own script, kind of your own understanding. I think we are going to, we recommended a analysis tool based on Python because that's the easier language to do. So you can create your own script. Like anytime you go deeper, you may have to create your own script to do something more specialized one. And you have to, but there, it's such a possibility to come up with new analysis techniques in MD. It is still, connecting the microscopic to the macroscopic world. There are so many opportunities, like it depends on your understanding. So it's a huge field. There is no recipe. You have to like follow other people, what they're doing and so on. And then one final thing I want to mention, another way of looking at the stability and as well as transition is called Ramachandran plot. Have you heard about anybody? Okay. Oh, good. So everybody knows Ramachandran plot. So okay. So in Ramachandran plot, you put the phi angle and psi angle. So in your protein, I think like N, Ca, CO, and the next N, right? So yeah, I think, let me see, if I'm not mistaken, this is phi angle. This is psi angle. I might be mistaken. Check it out. This is omega. Okay. So in a peptide bond, you have this chain ANCAC, then ANCAC, and so on. It goes like that. So let's put 1 before it would be C, right? So if you calculate these two, the plane that goes to these two molecules, and then plane that goes to these two molecules, and you see like how much are they rotated. That's your phi angle. Similarly, the plane that, so you need 4 atoms to, this is called dihedral angle. You need 4 atoms to calculate. Then if the plane that goes to these 2 atoms and the plane that goes to these 2 atoms, then you see like how much are they rotated. That's right. And you don't have to worry about omega. It's typically 180 degrees because this bond is very rigid. So that's why you only calculate phi and psi. Those are flexible. So you plot all the phi's and the psi's from your every residue. So for each residue, you're going to get 2 values, right? So this is one residue, NCAC, and then you have your residue side chain. That's one residue. So for each residue, you're going to get 1 value of phi psi. And as for each time step, you're going to get 1. So let's say I have, my protein has 1,000 snapshot. My protein is 100 residues long. So you are going to get 100 times 1,000, 100,000 values of phi and psi. Okay, and then you plot them like a dot here. Okay, well, let me plot. So in the Ramachandran plot, Ramachandran was one of the early Indian bioinformatician, really smart, should have gotten Nobel Prize, but didn't get it. He did all this work just by hand, stick and ball method. He moved something. He said like, these values of phi psi cannot happen. And these values of phi psi can happen. So then you have certain islands. And again, I don't remember which part is what. I think, yeah, so I think this is beta sheet. So if your phi psi values come from the residue within the beta sheet, they'll fall somewhere here. So it goes to minus 180 to 180, and then minus 180 to 180. I think so. Okay. And then this is alpha helix. Yeah, and then you have somewhere here probably 310 helix or left-handed alpha helix, I think. Yeah. So this is left-handed alpha helix. And then you have a 310, another kind of alpha helix that comes up and down here. So there you have some yeah, I might, it might be some, but you have some regions here, okay? So typically from your trajectory, you'll see most of your things falls here. And there are, because of the transition, you'll see like actually it is more like wrapped around, but most of it falls here and most of it falls here. Some falls here. If that is the case, that means your protein is behaving rightly. But if you see a huge spot in the middle of nowhere, that means like you have some problem. You have to go and check it. And due to some mutation, if you see certain aspects of it has changed, that's an effect of mutation. So that's another way. I mean, these are the initial analysis you do. And if you don't find anything into the initial analysis, then you go in deeper. And I think those all should be shown with your trajectory on the board. So you should have a, you should have a chance to look at how do they look. Any question? If not, let's see if we, well, any question? Yes.
Step 3, step 4, and step 5 of the simulation. Can you just in brief repeat one of them?
Yeah. So when we do a simulation, and this step 1, step 2, step 3, these are like charm GUI steps. Actually, it doesn't mean necessarily. So basically, what you do in charm GUI, the step 1 is probably reading the PDB. Step 2 is putting water and so on. Step 3 is maybe adding ions. I don't remember what is the step 3. What is step 3 is minimization. Okay. And in step 4, so up to step 3, everything is done on charm GUI website. The minimized step 3 is the minimization. In step 4, you bring it to your own MD engine, your own machine, right? You either run on MD or your GROMAX. So whenever you come at step 4, there will be certain minimization, and then you run it for certain step. We call it equilibration. That means you are letting the protein relax in that environment. And after the equilibration is over, then you start called production. That means like then you are going to run it for a long time, and that's where you are going to do your analysis and so on. Even when you are doing your analysis, not necessarily like you are supposed to take every part of the trajectory, because depending on what kind of data you're analyzing, the beginning of the step 5 trajectory, it may not have equilibrated enough. So you have to like plot it, and you say like, okay, after the equilibration, I'm going to analyze. So, but at least the step 5 is where you start your analysis. Yeah, you have a question.
Sir, I had a bunch of technical questions, to be honest. The questions, basically, I work at in silico predictions. So I make experimental designs, and if the predictions are correct or they are really accurate, my supervisor or teacher goes for in vivo validation. So if that works out, then we go for our publication. So I am the one who basically does the experimental design for the in silico part. So while doing this, there are many types of phytochemicals. We have worked on phytochemicals and other in silico products. The problem with MD is that what we faced when we wanted to validate our experimental, we know the results that this particular phytochemical or this particular toxic material might do this, might do that, and we have seen that. But when we come to do this in the in silico, we know the protein-protein interactions and all the mechanism of that. But we need to do MD to validate all those experiments we have done and all those mechanisms we have shown. So when we do that, let's talk about protein which goes to the cytosol from the blood and then delivers something, then it returns within 15 minutes. So if I want to show that in silico, I have to do MD, but I have to do MD twice. First, the protein was in blood, then the protein goes in endosome or cytosol. It has a different environment. Now, but the question is, but we are doing this on a particular toxicant, where instead of the product the protein originally delivers, there is another toxicant which actually binds to that protein, and that actually gets delivered to the cell. And that actually causes the problem. Now, if I want to see this, we have to, is that possible with MD? Can we see two different environments and two different products?
Yeah, that's actually a good question. I'll go through slowly. I'll replicate the question and you tell me if I'm replicating it right, and then you will answer. Okay. So okay, so you start with an experiment, right? So you have a protein, let's call it protein A. Okay, so this protein stays in the blood. Okay, so that means it's staying in extracellular environment, right? Yeah, so it is staying, and then what does this protein do? It delivers something inside the cell or inside the cell. Okay, so let's see. So this is my huge cell wall. Okay. And so this protein goes through the cell, and then where does it deliver? Okay, so it just delivers right inside somewhere. Okay, so it goes here, and what does it deliver? Yeah, so okay, so it goes through an endosome. That means like this creates a pocket. The protein forms here, and then this pocket of the protein goes inside. And okay, so what is the product? Huh? No, let's say that's okay. Just say like some more small molecule, like just generic. Okay, small molecule, that's fine. So you have a small molecule that is bound to this protein. Let's say we call this is bound to this protein, and is bound here, is bound here, somewhere here. It is get released because the pH is different here. So my small molecule is released, so I call it an S. So now it's released, and then it's probably released into the cell, right? Okay, so that's your system. And then what do you want to study with an MD?
The entire focus was published in a PhD paper back in 2021, if I'm not wrong, by an American scientist. The process takes around 15 minutes. The formation of endosome, then back into the blood again. It's an already published product, but we have seen it in a different way. That's why that oxygen got the particular different product and population of failure, that product comes in. So what we want to say that when it is in blood, it has a particular population. There is one inside. We know that we can relate it to the accident case, and then completely or slightly different environment than it was in blood. That actually that's the release of the product. So what we want to say that if that is true for the new thing.
Okay, so you have a different. Now what you are saying is this is the American version, so let's call it an SA, and you have a Bangladeshi version, let's call it an SB. So, and then whether does that do the same thing with an SB, right? That's what you want to do. So now if you want to replicate the whole thing in MD, forget about it. It's not going to happen. Okay. So so the first part is you need to experimentally determine where exactly, well, first question, does it produce the same effect phenotypically? It does not, right? So somewhere, wherever you put SB, is getting disrupted. Okay. So first, experimentally, you have to determine where it is getting disrupted, because otherwise you'll be throwing things in the dark. So, yeah. So the one way I would suggest, you call a fluorescence spectrometry, in the sense that you attached a fluorescent molecule to this protein, and you have access to SA, right? Yeah. So you attach a fluorescent molecule, and this is a very easy to see if it makes into the cell, if it does not mix into the cell, right? That will be your first step. So you see, like, does it do the endosome? First step. Okay. So after it, so this will tell you whether it gets inside the cell. Okay. So if it goes inside the cell, you don't have to do a run and MD. If it doesn't go inside the cell, then you come and build this and see what happens in the membrane, right? I mean, so that will be, okay. So now let's see if this one happens. Next thing is like your pH, and pH change is not hard to do with MD simulation. All you need to do is like change the protonation states, and that's your pH change. So if this one does, then I think like this can be actually done in your test tube, because you put in the test tube this system versus this system and change the pH, you should be able to see something whether this dissociates. I don't think you need to do an MD for this, right? Like if you want to, if you are looking for the answer, whether the pH change allows you to your thing to dissociate, you can do it in ITC, like thermal calorimetry. The thing is, like, if you change your pH, whether the binding and doesn't happen, or like if it get dissociates, you'll see it and signal in the calorimetry. So those are like more cheaper and more robust way of knowing, right? So and then finally, even if you find this one is right, then the next part is like whether this molecule, if it dissociates, whether it goes to the membrane of the endosome. Again, like, you know, so this is a too big of a picture. So you need to first break it down with experiment to pinpoint where you want to, and then you can ask the question to the MD simulation. Yeah. Yeah. Yeah.
Has this kind of thing, but not exactly. It actually forms the endosome while it actually forms an endosome while finding a homodimer. Earlier, you actually talked about homodimer a lot, then alpha folding and etc. We did that. We actually did alpha folding and other protein structure validation, but when that protein is actually not in the RCSB, not yet. The bigger protein is there, but the small protein which we are working on, that protein is not yet available. We are working on it in silico, and when we did the in silico part, everything went perfectly except for that. Every time we do the transmembrane protein, it doesn't go right. I have tried at least 10 different servers, from ITZ to GalaxyWeb to Coach to.
What does it mean it doesn't go right? Like what goes wrong?
The protein structure is different in every single server.
Oh, okay. So you want to first predict the protein structure of a protein that is transmembrane, and then you want to predict the protein structure due to a mutation, or just the protein structure due to a mutation? Okay. So I did not cover it, but can anyone tell, should we change a difference in a protein structure if it is mutated? If we predict the mutated structure using an alpha fold, so you have a wild type and an alpha fold, wild type alpha fold and mutated alpha fold. Should I see a change or should not I see a change? Answer meaning either way. I put the questions back to you. But okay, so you expect to see some change in the mutation, and so you're saying like if there is only single residue structure change, you should not see much change in the. Because you think like single residue is not going to change the structure by much. Okay. So what is the basis of AlphaFold? How does the AlphaFold gets the knowledge? No, multiple sequence alignment, right? So multiple sequence alignment means which two residues in history of the evolution are correlating, which two residues are in the history of the evolution are evoluting correlatedly, right? So I have residue 1 to residue 100, and then this is my protein 1, protein 2, protein 500 from different organisms, and similar residues, and I see like which one changes correlatedly. So in this thing, is there any mutation data? This is all wild type data, right? Because you don't have a history mutation, the one that you are looking at, if a mutation is stable, you move to a new organism, then it should be here. But the one that you are looking at is like probably a single mutation here, right? And if this mutation is evolutionary not stable, you are not going to get a signal from the multiple sequence alignment and correlation. So even if you put the mutation, the AlphaFold is going to give you the same answer. Okay, so the reason is getting all its data from correlated mutation over the evolution part. So that you have to remember that AlphaFold will give you the same answer. It's not sensitive to mutation. There are a little newer methods coming out. They're claiming it's sensitive to mutation in a similar machine learning. I haven't tested them yet, so I cannot vouch for them. Maybe if you look for the paper, they will come. So if you are trying to model a homodimer wild type versus mutation, you're going to get the same result. Yeah, so it's not, I think like if you want to do maybe MD simulation will be one way to do that, like wild type versus mutation. If you should be able to calculate the structure though. I think like, did you try AlphaFold? And it didn't? Okay. No, you just try AlphaFold, no other servers. I showed you the CASP, right? Yeah, so just AlphaFold, no other servers.
Part and just showed me the receptor protein, but the transformation protein was not there because the modeling or template-based modeling only had resemblance of the receptor, but not the transformation part. But without the transformation part, I cannot actually replicate my understanding.
Yeah, I think just stick with the
only AlphaFold and for mutation.
Oh, that's the whole, you have to run either MD simulation or maybe if you take some time to run Rosetta might be an option.
Yes, sir, I am learning Rosetta, but it's a difficult process.
It is a
very difficult process. I have tried Rosetta and it's not really that easy to master.
So Rosetta will be cheaper on the computational resource. You don't need as much. Maybe you don't need as much depending on, but it's a steep learning curve. MD simulation can be an option. Again, you have to define your question very well for an MD simulation.
I don't have the structure. I have to create a full structure to MD simulation, but I don't have that. AlphaFold won't give me the structure. If I do other stuff,
what do you mean AlphaFold don't give you the protein structure? Oh, you have to run the MD simulation and see. Yeah, you have to run the MD simulation with the protein, like without you take the wild type structure, right? You just change one residue and start with the wild type structure. And during the MD evolution, you see if it stays the same or if it changes. Yeah, you are not going to get any mutation. There is nothing that will predict you the server, predict the structure. There is just nothing there. And maybe some new one. I shouldn't say nothing. Like, yeah, there is nothing. Yeah. Oh. I think we need to take a break. What time is it? Oh, you're almost one, right? Okay. I think we need to go have, we'll continue this discussion. The only thing is like, are you done with downloading all the trajectory files? Okay, that's good. And I will download the Google, the Colab files. I'll download. You can download it too, and we can go over that at the board. Okay, and we'll continue this discussion. And I think, yeah, so if anybody has to go for namaz or prayer, we'll go, and then we'll go have lunch, and we'll leave. Okay, thanks.
আচ্ছা, যারা নামাজ পড়তে চাই তারা মনে হয় এখনই বেরোতে হবে, আর যারা নামাজ পড়বে না তারা ওই লাঞ্চ করে নিলে ভালো হবে। আমরা 2:15 থেকে 2:30টার মধ্যে আমরা অবশ্যই খুলনা থেকে কুয়েট থেকে চলে